Welcome to the SBGrid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SBGrid.
Hello, everybody. Welcome. I'm Pete Meyer from SBGrid. For today's webinar, we have Andrey Lebedev from the Science Technology Facilities Council in the UK. Just to give you a heads up for upcoming webinars, next week we have Yue Chang from University of Pennsylvania talk about cryo-ET reveals structures and potential regulatory mechanisms of rhodopsin secretion system in the apicomplexan parasites. And then the week after Thanksgiving, we have Yuntao Liu talking about ISONET, which is a deep convolutional neural networks for electron tomography. And you can send questions to the hosts in the chat and we'll moderate those at the end. And with that, I'd like to thank Andrey Lebedev for presenting today, and I will turn it over to him. So, show is yours. Thank you.
Hello, everyone. And I want to talk today about MoRDa pipeline for molecular replacement. It's one of many. However, there are several things to highlight here. Well, as any pipeline, it's automation, but also multimeric targets, and importantly, targets with novel conformations. And that is achieved by that MoRDa has its own database and associated programs that contains information about domains of selected entries from the PDB. Otherwise, it's a normal pipeline, which is a black box where you provide target sequence, X-ray data, and you've got solution at the end. We will look, the whole magic happens here inside this box in MoRDa-DB, and this will be the main part to describe what comes out of it into the molecular replacement and refinement block. Just overview, this database contains non-redundant protein chains derived from the protein data bank. And in this subset, the entries with high resolution are obviously preferred. And also there is a hierarchy of the chains derived from the PDB. There is also definition of about 100,000 protein domains contained in those chains. And definition also contain which part have to be removed and hierarchically organized according to the 3D similarity rather than sequence similarity. Multimers, if available in crystal structure, are also included, descriptions of multimers included, and those are generated using PISA results. Let's look closer what happens with the template search and model preparation. It's not terribly sophisticated, but importantly, it's all based on the domains. And first attempt is made, well, the sequence alignment is done first, and sequences of the domains are stored in the database. And then if any of the domains matches part of the sequence, that part is removed and a search continues until sequence is annotated by domains. Those domains then assembled back into the chain and also passed further down the pipeline as individual search models. Chains are passed down the pipeline as individual models, and also the multimeric models are assembled if there is a definition of them in the database. Downstream, we've got model correction using sequence alignment and secondary structure and surface accessibility. The surface accessibility defines the B factors that will be used in the molecular replacement search. Then all the models are grouped according to the PDB entries they are derived from, and we've got structure 1, 2, 3, and so on, as many as you defined in the command line or in the interface. each would contain sets of models. Some of the sets contain 2 or more models, and I will talk about it later. And there are chains and domains from the same PDB entry, and we'll just look at how it's defined. So domains in MoRDa database are not the same as biological domains, and some single domain molecules are represented by both, by chain and domain models. And the difference is that all flexible parts are removed from the domain models, but search is completed with both. One is more complete, the other is more precise. And here we've got a superposition of the target structure onto the domain model and onto the whole chain model. And it's in this example, domain model would work better. It's more precise. This is more or less the same thing, but in addition, well, no, sorry. Here we had a single domain model structure. Here we've got 2 domain structure. And now domain model essentially contains 2 models, which will be used in the molecular replacement search one after another. But they are grouped together, and there is an expected number of copies to be found for each. And we've got, again, chain models and multimer models, and ensembles, if possible, are generated. There will be one more slide about them. And in those multimers, in those ensemble models, domains from homologs are fitted into the domains rather than chains to chains or oligomers to oligomers. This allows for quite compact models, and at the same time big enough, bigger than a single domain. And at the same time, there are ensemble models which are known to behave well, perform much better in molecular replacement. Right. Let's look how that types of models fit in what you can find in the search summary tab of the MoRDa interface or MoRDa interface after MoRDa finished. So this is a search summary for whatever example that has been using for YZWA chain from the PDB. And that's PDB entry represented here by monomer and by domain models. And for each domain, information is given separately: identity, number of residues, and number of copies that are expected to be found. And that's yet another our example where we've got one PDB producing 4 different models, and actually we've got 2 models denoted by positive and negative numbers. It's just with swapped order. That only happens with the top 2 domains in size. But normally the search is completed with the bigger domain being used first, and I will explain later by. Well, it's obvious, but what it also implies in the molecular replacement. Right, that's easy one. Monomer and domain derived from one PDB entry with no actual domains, with a single domain. And there is a special kind of models that are sometimes generated, which contain domains from the different PDB entries. So here we've got PDB code, identity and number of residues, number expected, and ensemble model, whether it's generated or not. Recently, we have ensemble model nearly for all PDB entries, of course. And that's how this ensemble model are produced. Each selected model is converted to an ensemble model if there are homologs. And selected model acts as a reference, which shown green here in the picture. And homolog models are superposed onto the reference model domain by domain. And only the reference model undergoes alignment-based modification, surface bifactors modification, and contributes to packing function in MR, and is passed from molecular replacement to subsequent refinement. And that's schematic view of the same thing. Imagine orange is the upper, blue is the hollow protein, and there is also structures of their domains separately, and assembly model will look like as on the right. All the available homologs or close matches will be aligned to the confirmation that these two models represent, these two structures in PDB represent, and those two things will be tried in MR, as well as the domains derived from them. Recently, the different kind of model was added in which domains from different chains combined together as a single search model. And that is mainly relevant for the immunoglobulin-like structures, but sometimes it's also, well, it's not unusual also that we've got like dimerization or trimerization domains in the proteins, and those models would try, and those domains are usually oligomers, usually conserved, as like in helicases, whereas the other domains are in may take different conformations, so such models are good. And also they allow to solve automatically immunoglobulin-like structures, even if there are many copies in a symmetric unit. Now we go to molecular replacement search, and with the whole bunch of models, and we know that there are some so-called models that are actually several models that has to be run one after another. And structure solution is done using MoRDa and DriftMark, and Patterson search, conventional molecular replacement, is applied only to the first component in the series if that contains more than one component. And then it's only search in electron density maps is used for the second and further components of the complete entire search model. Partial structure is loaded in DriftMark, mark coefficients calculated, and the second search model and map coefficients are given to MoRDa, and that search is much more contrastant, as you can imagine, than the Patterson search. We've got already approximate phases there. In addition, search for the first component is done with trying different space groups if requested. Well, by default it is. The search is parallelized in the CCP4 servers, of course, and assembly search models, as I said already, only master model is passed to refinement. This is the molecular replacement solution, and subsequent refinements are represented by several entries in the log file. And in the short table, we've got number of copies found for either chain or domains composing it. We've got Q factor, which is a heuristic number, heuristic score based on the final and initial R and R3 factors, and that is between 0 and 1, and closer to 1 the better. And there are the stars that indicate whether the solution is likely to be a true solution or not, and space group in case if several space groups, if the space group is unknown and requested to try all, and the best will be shown here in the summary table. Let's also look at the two examples, which are quite nice because two examples of the models. One is the whole chain model, and it gave Q-score considerably smaller than the one for the domain models. And obviously it's because of domain flexibility. And so if we would use the whole chain, it's a slightly different example, but this sense is the same. If we would use the whole chain model, we would end up with the electron density maps as here, because domain is a different conformation. MoRDa strategy for chains will generate this one, but the MoRDa strategy for searching for individual domain will position the first one by search. Refinement will be performed, and the map for the second domain will help to put it much more precisely and actually in the correct position, so that the subsequent refinement will give a reasonable R factors and eventually Q-score for selecting this model as the top hit. There is also tab details that contains more information, including R factors, initial and final, and the score from the translation search, which is sometimes useful because it's not, it happens that it can be high, whereas the model wouldn't be decided to be the best solution. So it's worth to look specifically at that score if the case is problematic. Also here represented the solution in different space groups if the search in different space group was requested. Right. Now we are looking at the simplest thing, possible solution. It's just one section in the report page, downloads and everything, but it's not as simple in the other cases. We've got, as I mentioned, Multimer descriptions in MoRDA-DB, and that's one thing, but it's not necessary. But in case of multimers, not only multimers is being searched for, but the sequential search for individual chains is performed along in parallel with multimer search. Let's look at the examples with two sequences, and actually each of the chains contains at least two domains. And in that sense, it's an interesting example. So the report page looks differently now, and it contains more sections. First, we search for ensembles, and that's in parallel to the search in for the sequential search for domains derived from sequence 1 and sequence 2. And search for sequence 1, it would be looking same as in case of single sequence, same for the sequence 2, and we would have a best solution section displaying the history of the search of the best solution that has been selected as output from MoRDa. And we see that in this case, 5 domains were in one chain, and there were 2 domains in the other, which was bigger, or actually not. And nonetheless, this was quite easy to solve. Right. MoRDa is included in several interfaces. CSP4 has several. It's included in old CSP4i interface. It's also included in the new CSP4i2 interface shown at the bottom. Originally, MoRDa was written for CSP4 online services, where it's presented, of course. And we've got also a modern web-based interface called CSP4 Cloud that Eugene Krisnell will be talk about at SBGrid seminars next year, I suppose. And here we've got slightly easier. It's a little bit easier to handle because you don't need to see files or manipulate with files. All the data are passed along the branch in the project tree. And you can just see what happens looking at that tree. All branching and all successes are unsuccesses. Right, nonetheless, MoRDa is also a command line tool. You can run it like shown here in orange box, and it will do the same job, except that it won't be parallel as on the server. But also, and why it's interesting, this command line option, you can perform two parts of the structure solution process separately. First, generate models, maybe manipulate with them, or remove those you don't like, or add more models based on what has been already generated automatically, and continue to the molecular replacement solution as such. Now we will look a little bit what is behind the scene. We've got DB updates going on weekly, and that follows PDB releases, from which approximately, in average, 10% is being taken into the database, and those are entries that either have quite different fault or have quite big RMSD of the whole chain, the chain coming from the PDB. Multimer description are generated with Python, domain definition generated based on existing homologs, and that automatic definition then would be manually curated, and domain and chain trees are updated, and occasionally we do tests against the new structure, usually solved by SAT, and that to find out what we have to improve. And the whole new bundle, whole updated bundle of MoRDa and also the incremental tarballs go into several places from which they come to the user end, dependent on how MoRDa was installed, in what context. Most here are CCP4, but there is also a separate website which they are provided by BioMX. And so new PDB entries are processed weekly. However, updates to MoRDa and programs are released with intervals 2-4 months. They accumulate all the structures from the PDB during this time. Updates are automatically applied to MoRDa installation on CCP4 online and CCP4 cloud, but needs to be installed on local installation, and that can be done easily through CCP4 Update Manager if the MoRDa was installed with CCP4. Can also be applied to standard clone installation from the command line. There are 3 packages I'm aware of that use MoRDa as internal tool. That's Autoriksha, automatic structure solver at EMBL Hamburg. Simbat, which perform exhaustive search. It's no sequence molecular replacement, and it perform exhaustive search using MoRDa database. And that's also used for detection of contaminants, as well as there are cases of solving real new structures with this approach. And there is a contaminer that detects of contaminant crystals that also uses MoRDa. And here are the website where you can download CCP4, or download MoRDa. And the first one also contains a brief description how to use it. And that is it. Thank you very much for your attention, and I'm ready to answer questions if there are any.
Great. Thank you, Andrey. So again, if folks have questions, you feel free to send them in the chat or use the hand raise emoticon, and we can unmute you to ask yourself. So I guess a couple of questions. So one, could you say a little bit more about the surface B factor modification and what that entails and how it affects the process?
Surface B factor modification is, I think, commonplace in Phaser and MoRep. And the concept is that the surface is more flexible and less likely to be correctly determined in the model, and therefore additional B factors are added. What is done in MoRDa is purely heuristic, and I cannot tell any regular procedure that would give you numbers, really.
Okay, so this isn't doing anything like trying to match the Wilson B factor of the data set with the B factor of the search model, it sounds like.
No, nothing to do with this. It's not experimental. It's only about like prior information about it. It's not about data in the experiment. Errors in the experiment. It's about potential errors in the search model.
Okay, thank you. And I guess kind of feeding into that, what's your perspective on using side chains in the search models versus removing them depending on the quality of your data set? Is that always leave them in, always take them out, or somewhere in between?
I didn't say that. It's in the between.
Okay, you didn't mention, so it's probably why the question came up, I guess. So one other question. So with weekly MoRDa database updates, how do you recommend people cite that database, that snapshot that they used, or identify it when they're writing up their methods section? Is that just a timestamp, or is there another identifier?
There's the version. And if you look at the log file or report page, you will see the version there in Info tab. And that also contains the date when it was released. And that happens not every week, and we accumulate and then release.
Awesome. Thank you. I guess I apologize. It seems like I'm monopolizing the questions, but I do have one more, which is, and this is kind of more of an open-ended question, but would it be beneficial to use the results from earlier solutions to loop back and guide the selection of search models from the sequence alignments? Or is this a case where it's more go from the alignments and then use as many computational resources as it takes to find a good solution?
Here, there is no feedback from certain attempts to the next attempt. It's just deterministic, and its initial search is sequence-based, but subsequent selection is the RMSD based. So we've got lots of models selected initially, and then that's sequence-based. Apologies. Yeah. Then when we do ensembling, we do that. The RMSD estimation of what is the best match to this particular model.
Well, if it works without adding complexity, then there's no reason to add the complexity. So, All right, great. Oh, thank you very much. And I guess one final call for questions, and if not, then thank you again for joining us and for an excellent talk. So,
thank you very much. That was a pleasure. And
glad to have you with us and learn about the new stuff.